Disc 8.
The Human Genome Project has proved Darwin more right than Darwin himself would ever have dared dream. Molecular similarities stem ultimately from the way in which all organisms are related through common descent. A successful evolutionary invention, a mutation or set of mutations that is favored by natural selection, is passed down from one generation to the next. As the tree of life diversifies, existing lineages splitting to produce new ones. Reptiles persist as such, but also bud off into both bird and mammal lineages. That invention may eventually appear in a huge range of descendant species. Some 46% of the proteins we see in yeast, for example, also appear in humans. The yeast fungal lineage and the one that ultimately gave rise to humans probably split about 1 billion years ago. Since each has subsequently developed independently, free to follow its own evolutionary trajectory, there have been, in effect, 1 billion years of evolutionary activity since that yeast-human common ancestor. And yet, through all that time, that set of proteins that existed in the common ancestor has changed only minimally. Once evolution solves a particular problem, for example, designing an enzyme to catalyze a particular biochemical reaction, it tends to stick with that solution. We have seen how this kind of evolutionary inertia is responsible for the centrality of RNA in cellular processes. Life started in an RNA world, and the legacy remains with us to this day. And the inertia extends to the biochemical details. 43% of worm proteins, 61% of fruit fly proteins, and 75% of fugu proteins have marked sequence similarities to human proteins. Comparing genomes has also revealed how proteins evolve. Protein molecules can typically be envisioned as collections of distinct domains, stretches of amino acid chains that have a particular function or form a particular three-dimensional structure. And evolution seems to operate by shuffling domains, creating new permutations. Presumably, most new permutations are as useless as they are random, doomed to be eliminated by natural selection. But in the rare instance that a new permutation proves beneficial, a new protein is born. Some 90% of the domains that have been identified in human proteins are also present in fruit fly and worm proteins. In effect, therefore, even a protein unique to humans is likely nothing more than a reshuffled version of one found in Drosophila. There is no better demonstration of this fundamental biochemical similarity among organisms than so-called rescue experiments, the aim of which is to eliminate a particular protein in one species and then use the corresponding protein from another species to rescue the missing function. We have already seen this strategy implemented in the case of insulin. Because human and cow insulins are so similar, diabetics who fail to produce their own can be given the cow version as a substitute. In an example evocative of B-movie science fiction, researchers have been able to induce fruit flies to grow eyes on their legs by manipulating a particular gene that specifies where an eye should go. That gene then induces the many genes involved in producing a complete eye to go to work in that designated location. The mouse's corresponding gene is so similar to the fruit fly's that it will perform the same function when situated by the genetic engineer's sleight of hand in a fruit fly whose gene has been eliminated. That this can be done is nothing less than remarkable. Fruit flies and mice have been separated by evolution for at least 500 million years. So, following the logic applied above to humans and yeast evolving simultaneously along independent lines, The gene has, in fact, been conserved over 1 billion years of evolution. This is all the more astonishing when we consider that fruit fly and mouse eyes have fundamentally different structures and optics. Presumably, each lineage perfected an eye appropriate for its respective purposes, but the basic machinery for determining the location of that eye, needing no improvement, stayed the same. The most humbling aspect of the Human Genome Project so far has been the realization that we know remarkably little about what the vast majority of human genes do. To use the hard-won information properly requires us to devise methods for studying the function of genes on a genome-wide scale. In the wake of the HGP, 2 new post-genomic fields have duly emerged, both of them burdened with unimaginative names, incorporating the omic of their ancestors: proteomics and transcriptomics. Proteomics is the study of the proteins encoded by genes. Transcriptomics is devoted to determining where and when genes are expressed, that is, which genes are transcriptionally active in a given cell. If the genome is ultimately to be understood in its more dynamic reality, not as a mere set of instructions for life's assembly, but as the screenplay for life's movie, all the drama described in the precise order it is meant to occur, then proteomics and transcriptomics provide the keys to glimpsing the live action. The more we learn, the more we see of life, the movie. We've long appreciated that a protein is a great deal more in biological terms than the linear string of amino acids that compose it. How the string folds up to produce a distinctive three-dimensional configuration is really the key to its function, what proteomics seeks to know. Structural analysis is still done using X-ray diffraction. The molecule is bombarded with X-rays that bounce off its atoms and scatter in a pattern from which the three-dimensional shape may be inferred. In 1962, my one-time colleagues of the Cavendish Lab at Cambridge University, John Kendrew and Max Perutz, received the Nobel Prize in Chemistry for their elucidation of the structures of, respectively, myoglobin, which stores oxygen in muscle, and hemoglobin, which transports oxygen in the bloodstream. Theirs was a monumental effort. The complexity of the X-ray diffraction images they had to interpret made me appreciate the relative simplicity of DNA. Knowledge of a protein's three-dimensional structure greatly assists the work of medical chemists in their hunt for new drugs that work, as many do, by inhibiting protein functioning. In the ever more specialized and automated world of pharmaceutical research, several companies now offer to determine the structure of proteins as if they were production line commodities. And the work is now immeasurably easier than it was in the day of Perutz and Kendrew. With more powerful X-ray sources, automated data recording, and faster computers driven by increasingly clever software, the time needed for solving a structure can be reduced from many years